Right now, in some of you watching this, a virus may be living in your fat cells, changing instructions those cells follow. You caught it years ago. It probably felt like a cold, and then you forgot about it. This virus is tied to more body fat and better cholesterol at the same time. Wait, get fat and get better cholesterol readout? That shouldn't be possible. So I pulled the studies, including a 2024 paper that looked inside human fat. Did a virus help cause obesity? Let's read the evidence together. The virus has a name, human adenovirus 36. It lives in the same family as the viruses behind common colds and pink eye. I'm Dr. Amin Hedayat. I'm a triple board certified physician, pathologist, and assistant clinical professor. As a specialist studying human tissue, this headline surprised me. Scientists gave the idea its own label, Infectobesity. The claim is that a common infection can reprogram your fat tissue towards storage long after you cleared it. If you've struggled with your weight and suspected the usual advice was missing something, the possibility matters and you deserve a no-nonsense answer. In this video, I'll be evaluating the credible studies and I'm grading it. Solid means the data is strong. Emerging means the signal is real, but so are the gaps. Unproven means the story ran ahead of the proof. I try to grade studies and claims on this channel using the 3 steps. If you enjoy actual facts without hype, please subscribe and hit the notification bell so the next deep dive video comes up on your feed. We'll do this video in this format so it makes sense. Animals first, then the mechanism inside the cell, then people. Each step gets a verdict. Please don't skip ahead so the progression makes sense. Let's start with the animals because that's where this began and it's the strongest ground we have. When researchers infected chickens, mice, and even monkeys with adenovirus 36, the animals got significantly fatter and it repeated across species. For animals, I graded solid. Then the unusual part, those fatter animals didn't have worse cholesterol. Theirs in fact dropped, which is why this virus is so interesting. So why would a virus grow fat tissue? Let's think about the architecture. Your fat isn't a passive storage tank. It's a living structure with a workforce. Some cells are mature and full. Others are quiet, stem cell-like cells called pre-adipocytes. Picture them as empty lots, zoned for building, waiting on instructions. Adenovirus 36 carries one gene that acts like a foreman walking onto that site. It's called E4-ORF1. In cell studies, that single gene does 3 things. First, it wakes up the empty lots. It pushes pre-adipocytes to become full fat cells. New storage units come online. The tissue doesn't just fill the cells it has. It builds new ones into the cellular matrix. Second, it flips the master switch for fat cell formation, PPAR gamma and CEBP. Turn those up and the whole building program runs hotter. Third, and this is the clever part, E4-ORF1 pulls glucose out of the blood and into the fat cells. It opens the doors that let sugar in. Now the paradox makes sense. If the virus is shunting sugar into fat instead of leaving it in the blood sugar can read lower while the fat quietly expands. More storage and you have better looking labs. That's exactly what showed up in the animals, and that's why some researchers have studied the same gene for the opposite reason, as a possible tool against high blood sugar. So this one mechanism has 2 faces. In cells, that mechanism is solid. Knock out E4-ORF1 and the effect disappears. Put that one gene in by itself and fat cells form, making the gene necessary and sufficient. What I won't do is assume a chicken and human run identical wiring. So let's talk about people. In 2005, a team led by Dr. Richard Atkinson ran the study that put this on the map. They tested 502 people for antibodies to adenovirus 36. Antibodies are basically the immune system's memory paper trail. You carry them because you met the virus at some point in your life. Adenovirus 36 antibodies were identified in 30% of people with obesity and 11% of lean people, nearly a 3-fold gap. And it held independent of age, sex, and where the sample came from. Now look at that number again, 30%, which means 70% of the people with obesity in that study showed no sign of the virus at all. Whatever built their fat tissue, adenovirus 36 wasn't even there. And look at the lean group. 11% of them carried the antibodies. They met the virus. They stayed lean. So no, adenovirus 36 doesn't appear to make everyone obese. It isn't a switch you either flip or don't. At most, it's a thumb on the scale. One input sitting next to genetics, sleep, food, medication, and everything else in a life. Why it seems to push harder on some people than others, I don't know. Nobody does yet based on the published research. Antibodies tell you the virus visited. They don't tell you what they did while they were there and whether it ever reached fat tissue in that person at all. So that's the first limit. Let me tell you about the second limit that's even more important. An association isn't a cause. Maybe the virus drives the fat. Maybe carrying more fat makes you easier to infect. Correlation runs both ways. So the same team did something smarter. They looked at twins. Twins share a womb, a household, and the same food growing up. That strips out an enormous amount of the noise. In pairs where one twin carried adenovirus 36 antibodies and the other didn't, the antibody-positive twin had a higher body mass index and more body fat than the other sibling. In other words, same household conditions, but different antibody status. Measurably more fat in the one who met the virus and had the antibodies. That's stronger than a population count. It's not proof, and the numbers were small, but it's the kind of result that makes you keep looking for a more precise answer. This is a fair spot to pause. On my 3 stamps, where would you put this so far? Drop solid, emerging, or unproven in the comments. I'll give you mine in the end. Your engagement really sends a positive signal to the algorithm that you're following the video and engaging with it. Also, I would appreciate you liking the video and sharing it if you find it interesting. So back to the topic. So the paradox held up in people too. The antibody-positive group had lower cholesterol and triglycerides, the same pattern as the animals. More fat, better lipids. That consistency across species is what lifts this above ordinary hype. But notice what every human data point so far has in common. It's a snapshot from the outside. Antibodies in blood, weight on a scale. To take this seriously, you'd want to look inside actual human fat cells and ask whether the virus left a fingerprint on the machinery itself. For years, nobody had done the study cleanly. In 2024, someone did. Before I share with you the 2024 data, a quick 10 seconds. Everything I audit on this channel is part of a step-by-step system inside the Get Uninflamed platform. The app, a graded video library, tracking tools, a real community. The founding cohort opens October 15th with the GLP-1 method. The waitlist enrolls first, and you can waitlist for free at getuninflamed.com. Now back to the tissue. In 2024, a group publishing in the International Journal of Obesity did the thing the field needed. They took visceral fat biopsies straight from people during surgery and compared the tissue itself. 2 groups, both with obesity. One carried adenovirus 36 antibodies, one didn't. Then they studied the fat at the molecular level. Meet 2 players, GAS5 and MEG3. I'll call them GAS5 and MEG3. They're long, non-coding RNAs. Don't let the name scare you. Think of them as safety governors built into fat cells. Their job is to slow fat cell formation. They're the brakes on the building program. So what was the finding? In the people carrying adenovirus 36 antibodies, both brakes were turned down. GAS5 significantly lower. MEG3 significantly lower. The governors that normally keep fat tissue from overbuilding were quieter, specifically in the people who had met this virus. At the same time, a small molecule called MIR18A that pushes fat cell formation forward was higher, and GAS5 tracked almost perfectly against PPAR gamma, that master switch from earlier. As the brake fell, the switch climbed. This isn't an antibody floating in blood anymore. It's a change in the actual control machinery inside human fat, matching the exact mechanisms we saw in the lab. The architecture story and the human tissue finally lines up. So do we have our answer? Did a virus make you get fat? No, and I won't dress that up. That study looked at roughly 57 people with obesity. That's small. It's a snapshot at one moment, not a film of what changed over time. And it still rests on antibodies, which only tell you the virus visited. They don't tell you the virus flipped those brakes or whether some third factor flipped both at once. There's a deeper wall. To truly prove cause, you would have to infect people on purpose and watch what happens. but you can't. It's unethical, and it won't pass the ethics boards. The clean randomized trial that would settle this can't run in humans. So how does the rest of the literature look? A 2021 systemic review pulled 37 studies. 31 found the association. 4 found nothing. 1 found the opposite, that adenovirus 36 looked protective. So the signal is there, but it varies widely. Some groups show about 5% with signs of past infection, while others are near 50%. And since labs don't run antibody tests the same way, the results are hard to compare. 31 out of 37 sounds like confirmation and proof, but in reality, it isn't. It's 31 snapshots, and not one of them can tell which came first. The effect may be real, but it's modest, tangled up with genetics, diet, and environment. In other words, there's no one clean cause, which is exactly what those 70% were already telling us. So my grade on the headline that adenovirus 36 causes human obesity, it's unproven. The cell mechanism, on the other hand, is solid. The human association, it's emerging. Human causation is not established, and anyone who tells you otherwise, stop reading at the abstract or is trying to sell you something on hope. This is a fair audit, strong where the data is strong, and I'll be honest about where it's not. So let me close the file. Adenovirus 36 makes animals fatter. It's solid. It carries a gene E4-ORF1 that drives fat cell building in the lab. It's a solid. In humans, linked to more body fat in some people with the fingerprint now found inside fat tissue, it's emerging. That it caused your obesity, it's unproven. If you came in hoping this virus was the answer, the one thing you could finally treat, I understand the hope. There's no approved adenovirus 36 test, no antiviral for weight, and don't buy a product promising to end the root cause of your obesity. But every study we walk through points in the same direction. Obesity is biology, not verdict on your character. Your fat tissue runs on instructions, and instructions can be rewritten. The switches this virus reaches for, PPAR gamma, lipogenesis, inflammation, also answer to what you do every day. Sleep, protein, resistance training, fiber, less ultra-processed food. None of that is glamorous, and none of it is a moral test you've been failing. It's leverage over the same machinery the virus was pulling on. You can't go back and audit a virus from years ago. You can, however, protect your baseline starting tonight. That's always more in your hands. If you enjoy more deep dives like this in your feed, please share topics that interest you and make sure you're subscribed. I read these comments, and your comment may become my next topic. If you're interested in the deep dive on the peptide stacks that are very popular these days, I'll leave a link on the screen that will take you there. Also, don't forget to tell me in the comments where you landed on the 3 steps. Thank you for watching this video. Stay uninflamed, and I'll see you in the next video.